Buonasera.
Hi Sebastiano.
Hi to everyone. Hi Pietro, hi Edoardo.
Hello everybody.
Well, I guess I'm free.
Okay, I think we can start very much on time since we have a lot to cover for today. And welcome to everybody to today's webinar. My name is Edoardo Bertero, and I will be the moderator for this session together with my colleague Pietro Ameri from the University of Genova. And today's topic is cardioimmunology, which is a very interesting and expanding field of research. And we have three outstanding researchers that will guide us through very different areas of the interaction between the immune system and cardiovascular system. We also have two outstanding discussants that will lead the discussion at the end of the webinar, Cinzia Perrino and Sebastiano Sciarretta. And I think we have a lot to cover and we need to stay on time. So without further ado, I will hand it over to Pietro Ameri to introduce the first speaker.
Yes, sure. But well, first of all, very nice to see you all. It's good to see that there are many people connected. And our first speaker is Wouter Myers, who's now based in Rotterdam at the Erasmus Medical Center. Probably many of you already know him because of his interest in immunocardiology, especially immunocardio-oncology. And we are just very much looking forward to listening to you. Wouter, please.
So, good afternoon, everyone. First of all, it's an honor to talk to this Italian crowd here this afternoon. And of course, Pietro, Eduardo, thank you for the invitation to share a bit about the research we did and are currently doing, because there's a lot of developments going on. I'm trying to share my screen. Let me know if you guys can see it and see the presentation.
Okay, we see your screen.
Perfect. Then let's get started. So the first talk is about the immune checkpoint inhibitors and related toxicity, and maybe beyond the toxicities, and in the 9th of October in Italy. These are my disclosures. I wanted to start off with a patient case because I think this is a mixed crowd that we have, and it's an old guy born in 1924 with not a really typical medical history, because he collapsed in 1917 while running, but also had a tear gas in the eye in 1977. And I know a little bit more about the details because he had hemorrhoids during the Christmas party where he needed to leave early. But he also won the Nobel Prize in 2002. And of course, the Nobel Prizes are handed out as we speak. So he got one in 2010. But he was unfortunately diagnosed with a malignant melanoma in 2015, with also metastases to the liver and the brain. And luckily for him, this was right around the time that these immune checkpoint inhibitors came across. So he was one of the first patients really treated with these inhibitors. And 1 year later, he was declared cancer-free. And of course, of an old age, he's now in hospice care at the age of 99, and his wife passed away recently. And I know this case very well because of all the presidents of the United States, and Jim Carter, his medical history is known. So he's still living today, but he was one of the survivors of metastatic melanoma, which of course is breathtaking to start with. How did it all came across? This is Jim Ellison from the US. And around the time in 1995, he was finding out the mechanism, how these immune checkpoint mechanisms work with CTLA-4 and CD28. And he really saw they have opposing effects with simple assays, really looking at T-cell activation, with higher CTLA-4 putting it down, but higher CD28 putting it up. So very easy experiment and showing that he could manipulate T cell function. During the same period, on the other side of the world, in Japan, we had Hanjo working on PD-1. In the biopsy mouse model, he saw that the PD-1 knockout, so if you have no PD-1 in your heart, leaded to a dilated cardiomyopathy. And he published this in Science in 2001 without even knowing what caused it. And in his follow-up experiments 2 years later in Nature Medicine, he saw that it was against troponin I and an autoantibody due to this PD-1 knockout that causes this dilated cardiomyopathy. So both won the Nobel Prize, I think like 5-6 years ago, focusing on this immune checkpoint inhibitors PD-1 and CTLA-4. What is now the exact mechanism? And I think you all know this pretty well, so I will keep this short. We have the T cell and the tumor cell, and the T cell recognizes with the TCR recognition bindings that it is a tumor cell. The tumor cell actually wants to survive and don't want to be killed by the T cell. So this is where this PD-1, PD-L1 connection leads to T cell inhibition. So this is a protective mechanism of tumor cells. And this is where this immune checkpoint inhibitors come into play, because then they break this inactivation, so the T cell becomes reactivated again, and then the tumor cell. This is in a nutshell how these mechanisms work. And if we look at the clinical trials that were being conducted, this was metastatic melanoma, 40% survival after 12 months. And this was only one immune checkpoint inhibitor, because oncologists go crazy nowadays with different combinations, but they saw a very nice improvement and also durable response. And it was better tolerated than even chemo. Then, for example, immunologists, they can very nicely look at the tumors itself. They did stainings, like how much of this PD-L1 is present. And then based on the presence on the tumor, so really targeted therapy, they say if you have a high presence, you might have better responses. And it was also the case for in these cancer types. And this led to a free fall. The industry jumped on it. We did a lot of studies in a lot of different kind of tumors and with different kind of combinations, chemo in combination with immune therapy, but also different kind of immune therapies combinations. And this is a little how it looks like up until now. So we recognize that PD-L1, PD-1, CTLA-4, and LAG-3 are the most common targets nowadays and in different types of cancers. And there is still so much clinical trials being conducted as we speak. But with great power comes great responsibility, because we see a lot of toxicity due to these kind of treatments, and we see them in the entire body. And as you can see, all of these diseases end with itis, because inflammation is the common theme here. And unfortunately, also myocarditis. And I want to zoom in on that a little bit more, because if we look at ICI-mediated myocarditis, if you look at the left, you see the incidence, you can say, oh, the incidence is pretty low, roughly maybe 1-2%, but the fulminant cases have a very high mortality, ranging to nearly 40%. And if you look at the clinical phenomenon that we see, if you focus on myocarditis, this is early on in the treatment. So the right of the x-axis shows how many infusions people got, and really after 1 or 2 infusions, patients normally present with myocarditis. Then everybody wants to know who's at risk. And we also try to figure it out with large databases and meta-analysis to come up with treatment. And we see it more happens in females, and if people start combining drugs. So combination immune checkpoint therapy or with targeted therapy, these patients are more prone to have ICI-mediated myocarditis. Then, of course, we didn't know where this was going. So I think it's already like 2-3 years ago, we came up with this preclinical ICI myocarditis mouse model. It was like a combination of a PD-1 knockout and an happens to call CTLA-4. So the combination was dropped. And you see this nice in red, 50% of the mice survived and 50% died, made it the same as we see in humans. And if we look under the microscope, this is how human myocarditis looks like. And in the murine samples, it was the same. These mice were dying of myocarditis. And if you look at cardiac infiltrates, you see half of them also clearly show this. And then troponin I and measurements, troponin I, a measurement that we're all familiar with, we also saw in half of the population being elevated. So we really had a model here that we could study the disease more clearly. And then, of course, we went into immune. I don't have to, with all the crowd here, we don't have to dive in, but you know CD4, CD8, and the macrophages, what was driving this process. And we easily found out that if you get rid of all the CD8s, you could rescue the phenotype. So we came up that at least in this model, and we also believe that's in human case, it's a CD8 phenomenon that we see with ICI myocarditis. But in humans, it's very hard to get rid of all your CD8s because then you will be prone to a lot of different kind of diseases. So are there druggable options? And then abatacept, this is like a third line or a fourth line treatment in rheumatoid arthritis, came up. It really has a nice interaction with CTLA4 and CD8. So it's a CD8, CD86 ligand. And it especially functions very well in activated or deactivated T cells. So we tried it in our mouse model as well, and it had a phenomenal result with saving this and rescuing this phenotype in the mice. And we also looked at immune infiltrates over time. You can clearly see, not at the beginning, but really after 14 weeks of treatment, abatacept had a beneficial effect. Nowadays, this Erasmus MC is the Rotterdam hospital that I work. This is a little bit how we treat patients with fulminant myocarditis with medical treatment, because some of them unfortunately end up in the ICU needing mechanical support. But everyone is started on high dose of methylprednisolone for 3 days, and we try to taper it down thereafter. And our second line of defense is more like our transplant rejection kind of therapies that we infiltrate. You have to realize if your patient is not responding well, these oncology drugs, they stay in the system for 6 weeks. Something to realize. So this is an ongoing threat. So this is why plasmapheresis comes into play. You can do targeted immunoglobulins, but also now we had 1 or 2 cases where we needed to go to these abatacept, for example, as a fifth line of treatment because there was nothing else to do. You were standing with your with your back against the wall, so you needed to go in these kind of things. But this is a little bit the regimen that we do on medical therapy in patients in our hospital. And I still have a little bit of time left because I want to tease also a bit where this field is going, because, and I think also maybe the next speaker will maybe dive into this. T cells and immune checkpoints might be also important in other cardiovascular biology, where T cells come into play. And for example, it can also play a role in non-ICI myocarditis, but maybe also, for example, myocardial infarction, because we all know at the beginning of this disease, there's a lot of immune infiltrants, and we see, for example, now with treatments of colchicine and these types of drugs that they have beneficial effects. So the question is, does immune checkpoints also play a role in other cardiac diseases? And the interesting thing is we did a few studies, and this is one study where we looked at immune checkpoints as a biomarker. And these are 2, I will show the first one first. It's a chronic heart failure cohort. And here you can see 3 of these immune checkpoints. And we saw just with hazards, just simple statistics, if they predicted heart failure hospitalization and all-cause mortality. And you can clearly see a signal that if you have these antigens floating around in your system, you're more prone to develop adverse events. And if we look at a very worsening, more acute heart failure cohort, more patients, more power, and PDL2 and galectin-9 were present, we really saw that if you have, so these are oncology-driven biomarkers, right? Because it came from oncology. They have at least a signal also in the field of cardiovascular disease, and in this case, heart failure. If we zoom in, this is unpublished data. We did some interesting findings in the myocardial infarction cohort. So these are patients with their first myocardial infarction going for PCI in our center. And we did 4 months later follow up with MRI to see how the infarct size was and the LVEF. And nowadays it's the same as in Italy. It's like within 1 hour, people are getting these PCIs after they have chest pain. So you don't really see that much of difference with LVEF over time in the complete cohort. But what was interesting is that these are TIM3 ligands, is also an immune checkpoint. You could clearly see that they were associated with the infarct size. So you could say maybe the inflammatory process would also yield to maybe fibrosis formation and is associated with these immune checkpoints, and it also correlated with LVEF over time, 4 months later after myocardial infarction. But interesting, the same idea is that something comes from oncology field and can be implemented maybe in the cardiovascular field. And research you don't do alone. And so we work with a great group in Erasmus, and we also have like shared collaborations with Italy, Pietro, of course, and Raquel, who also joined this meeting. And of course, with my ongoing, where I did my postdoc, and a very good combination with the Budapest group. So thank you all. I'm open for any questions, or maybe we save them for the discussions afterwards. But let's hear from the moderators.
Yeah, thank you, Wouter. Yes, discussion will be at the end, but thank you for now for this excellent presentation, a very nice story of translational research, showing us how you can go bench to bedside first to treat cancer, and then back to the bench again to treat the toxicity, and ultimately leading to some discovery in how heart failure and cardiovascular disease progress in the interaction with the immune system. And I will move then now to our next speaker, who is Dia Ashur from Germany, from Würzburg, Germany. He is an expert in immunology. He earned his PhD at the University of Würzburg, and is now a postdoctoral fellow in Gustavo Ramos' laboratory, where he investigates the mechanism of T cells and the role of T cells in cardiac health and disease, which will be the subject of his presentation. So please, Dia.
Thanks a lot for the introduction, Eduardo, and thanks a lot for the invitation. I'm really happy to be here today. So I would share my slides. You can see my slides, right? So yeah, so the topic is rather a bit broad, yeah. We start from health. We're mainly focused, of course, on myocardial infarction in the lab and also on aging. But let me walk you a bit first through how the T cells look like in a healthy heart. So this is like a 3D representation of the immune cells of CD45, immune cells within a healthy mouse heart. And perhaps what you can see, what strikes you at first is that they are not the most abundant population of cells in a healthy heart, yeah? And if you move on to a human data set, this is from the human heart atlas, and you just see T cells compared to the other immune cells that are residing in a, well, physiological condition of a healthy heart, also in humans. You see that the situation is rather subtle. Yeah, you don't see many T cells compared to the other cellular subsets that are present in the heart. Now, the situation changes a lot during cardiac pathologies, and as Dr. Myers said in the last talk, during autoimmune myocarditis and also ICI myocarditis topic now, I would say the T cells are considered kind of initiators of disease. They are more described now as initiators really of the cardiac pathology, where they are saying that cardiac antigens presented by these antigen presenting cells and propagating the disease pathology. In other cardiac diseases, for instance, in HFrEF and also now in HFpEF, they would be considered more of amplifiers of the disease, and modulating them would rather be to tone down the disease progression. And then we have ischemic heart diseases, where they are more described as rather modifiers of disease. So depending on when you look, they can be more causing repair mechanisms for the heart or inducing more collateral damage in the heart. Now, the kinetics of how the immune cells infiltrate into the heart during ischemic injury has been quite well described. Right after the injury, the first immune cells that infiltrate into the heart are neutrophils. They're important for clearing up the debris of the dead radiomyocytes. And next, start an inflammatory phase where the main cellular subsets that are influxing into our heart are monocytes and macrophages that are rather with a pro-inflammatory phenotype. And then they slowly get replaced by also macrophages, but that are rather having a pro-healing phenotype, and they are cross-talking a lot with the fibroblasts that are important for building the extracellular matrix. And at this stage, this proliferative phase is also when the T cells start to kick in together with B cells. But we have to keep in mind that this is again single cell sequencing data from mouse post myocardial infarction, experimental myocardial infarction, that if you compare, let's say, a day 3 during the inflammatory phase, the monocytes and macrophages compared to T cells, the T cells are really a minor population. And then if you go look at day 7, where there's kind of like the peak of T cells, they are still rather a small population compared to monocytes and macrophages. However, just like the number of cells does not mean the function of the cells. And one important aspect of that in the T cells is that they don't do all the job in the heart. They rather see the signal, which is the antigen presentation by the dendritic cells within the lymph node that drains the heart. And then they get activated here and go back into the heart to do the job. So in mice, of course, we can look at these mediastinal lymph nodes that are draining the heart. However, what is the situation in humans? So one previous study in the lab did CXCR4 PET tracing in MI patients compared to healthy cohorts. So this CXCR4 is a molecule that is present on different immune cells. However, in the lymph node, the main population of immune cells that will be expressing this would be the T cells. And you can clearly see that after myocardial infarction, the lymph nodes of these patients are increased in size and they shine more for this marker, indicating that there's definitely something going on in these lymph nodes with the T cells post-MI. Now, it's also important to know that T cells are quite complex in their biology of how they recognize a dangerous signal in our body. So, and this is because this is inherent to how this T cell receptors of them are generated. So the T cell receptors, they are formed during the development within the thymus in a series of complicated recombination events between several junctions that are in the genome that are basically cut and recombined for both the alpha and the beta chain of the TCR. And this gives a potential that it can basically recognize any kind of antigen that is present. And the theoretical numbers for how many TCRs can be generated is up to 10 to the power of 19, which is, of course, a lot more than what the body can have this number of cells. So I just wanted to give you this brief introduction because there is already a tool, a nice tool that was developed by Professor Bucher-Ludwig from St. Gallen, which has, so this mouse model has these so-called TCRM cells, which carry a T cell receptor that is specific to a dominant myosin antigen. And it was mainly developed to study myocarditis. And these mice develop myocarditis spontaneously. However, we can also use this mouse model, of course, for other cardiac pathologies. And first, what we did was a screening for the different peptides that elicit an immune response or a T cell immune response post-MI in experimental MI models. And we see that one dominant antigen that is being present is this exact same epitope for myosin. And if you transfer these cells from this TCRM mouse before ligating the coronaries of the mice, you see that these cells home into the myocardium. Now, Murillo from the lab also did further phenotyping of these T cells, and you can clearly see that during the inflammatory phase, so around day 5 to day 7, these T cells acquire a more of a surprisingly regulatory phenotype. They home to the heart more and they acquire a regulatory phenotype. And I won't go too much into detail, but these UMAP plots just show you basically that these TCRM cells are having a more of a T regulatory signature and downregulate their pro-inflammatory gene status. And if you pre-differentiate these T cells into a T regulatory phenotype before you inject them and then induce myocardial infarction and compare this to TCRM cells that were not pre-differentiated, you see that the myocardium adopts a more like downregulation of pro-inflammatory cytokines and upregulation of extracellular matrix remodeling genes. So all in all, it seems that at least at this time point, these myosin-specific T cells, they foster more pro-healing and better repair mechanisms in the myocardial infarction. Now, this is just one thing that we're looking in mouse models that are young and healthy. However, when we look in the clinics, most of the people that are presented with ischemic heart diseases, they are rather elderly, and probably their immune system and their myocardial system and their cardiovascular system has undergone a lot of changes during their aging process. And this is probably an intertwined process where the T cells are undergoing more senescence because of being exposed to chronic infections. And this goes along with reduction in myocardial repair processes and an increase in heart failure progression. So we and others have seen that just if you look in healthy mice that have aged compared to young mice, and you look at different cellular subsets within the myocardium, you see that inflammation is going up, basically. And this is termed inflammaging. This has been a term that has been coined since 2001. And if you look at some of the canonical inflammatory markers, for instance, TNF, you see that this is happening not just in the heart, but also in other organs along the aging axis. Now we try to understand a bit more how this T-cell myocardial crosstalk is happening during aging, and for that we serially sampled T-cells, activated T-cells from different age groups from the mice, and simultaneously looked at the hearts in 2 months and 18 months of age. And we specifically looked at the non-cardiomyocytes because they are hard to look at with sequencing. And basically, from the T cells, from the mediastinal lymph nodes, we see that there's one prominent cytokine that is building up along the aging axis, which is interferon gamma, which is a major pro-inflammatory cytokine for T cells. And this goes along with senescence signatures and more T cell receptors that are expanding, probably to an antigen we've yet to understand. Now, this goes hand in hand with an increase in this signature for T cells that infiltrate the heart. And if we look in our data set and probe also other publicly available data sets for aging of the myocardium, we see that an interferon gamma response signature is building up. So there seems to be a crosstalk along this interferon gamma axis between the T cells draining into the heart and the myocardium. Now, what was interesting is that this interferon gamma response is mirrored with metabolic shifts that are quite reminiscent of what's happening in failing hearts. So we see a downregulation of oxidative phosphorylation, though also glycolysis. We tried to at least recapitulate that in vitro first. So using iPSC-derived cardiomyocytes, if you put interferon gamma on them 1 or 4 weeks and then do seahorse assays, you see that oxygen consumption rate and extracellular acidification rates are going down, indicative of, blockade of OXPHOS and also downregulation of glycolysis. And although this is still ongoing, what we're doing right now is to try to recapitulate this system in vivo. So we have an adeno-associated virus delivering interferon gamma to mice. And if you look 28 days post the delivery of the virus, we see a similar phenotype as to what we saw with aging and what we saw with the iPSC-derived cardiomyocytes. So the myocardium itself is pushing more this interferon gamma response, and this goes along with metabolic shifts again, like oxidative phosphorylation downregulation and fatty acid metabolism downregulation. So we think, of course, we're still touching, the tip of the iceberg. I think it's really important that we try to understand more how this cardioimmune metabolism is happening along the aging axis, and try to address more that we get a balanced immune response, that the T cells are really within the right phenotype to induce a proper healing phenotype. And of course, the big question of what antigens are these T cells seeing, so that we can harness this in a more personalized medicine. Okay, with that, I would like to close. I would like to thank all the people from our lab and all our collaborators, and thank you all for your attention. Thanks.
Thank you so much, Dia. Very, very nice talk. So we'll keep on with the schedule, and we'll have the last talk, and afterwards we'll discuss all the three speeches together. So the next speaker is Professor Daniela Carnevale, who's full professor at the University Sapienza in Rome, and she also leads a research laboratory dedicated to cardiovascular immunology. And today she will talk about the neurovascular interfaces in atherosclerosis. Please, Daniela.
Thank you for the introduction, and thank you for the invitation to this nice webinar. As introduced, my talk will deal with neuroimmune interfaces, and in particular with interorgan communication in cardiovascular disease. And before starting, I would like to introduce two key concepts that I believe are really important in the setting of interorgan communication. One is the so-called interoception. The interoception is the way our brain perceives everything is happening in our peripheral organs. And this is a typical representation of the mouse brain, where there are highlighted some areas that are typically responding to peripheral stimuli. And another newer concept is the so-called immunoception. Immunoception is the way our brain can map what is happening in our peripheral immune and inflammatory responses. This is an example of how some neurons in the brain are activated, switched on and off during peripheral inflammation and immune responses. Why these two concepts are important? Because these two processes are the processes that allow our brain to be in constant contact with our peripheral organ and establishing bilateral communication. And how our brain can establish bilateral communication. Typically, our brain is able to use what we call the peripheral nervous system, that is made by different array of nerves that bilaterally communicate between brain and peripheral organ. Here there is a typical representation of the brain communication with peripheral organs. And the most important and the newer thing, because this is something that in cardiology we know from decades, but what is more interesting now is the fact that the spleen and bone marrow, and in general immune organs, are densely innervated by our peripheral nervous system, creating an array of nerves that go bidirectionally between the brain and the immune system. These are typical representation of primary and secondary lymphoid organs that receive a different type of innervation from the brain. And you can easily see that there is always an innervation by the sympathetic nervous system, suggesting that when we have an enhanced sympathetic outflow, that is a typical condition in cardiovascular disease, our immune system is activated. Our lab some years ago now decided to find a way to directly measure how the brain controls the immune system during cardiovascular disease. And by taking the advantage of the typical electrophysiological method to record the peripheral nerves, the microneurography that was used in the past for measuring renal nerve activity or muscle nerve activity, we changed the system a little bit to measure splenic nerve activity. The splenic nerve is a tiny noradrenergic nerve that connects the celiac ganglion to the spleen, and is a way the brain uses to control immune system in the spleen. In different models of hypertension in the past, we found that basically there is an early surge of sympathetic nerve activity on the spleen. This is the model of angiotensin II. This is a salt-sensitive hypertension. This is an obesity-induced hypertension. In all this model, we have a surge of the sympathetic outflow on the spleen, suggesting that there is a rapid reaction of the sympathetic nervous system to these stimuli to modulate the immune response in the spleen. And when we looked at the molecular mechanism inside the spleen, we characterized the release of a growth factor that we have characterized over the past year, and it's called the placental growth factor, because it belongs to the family of vascular angiogenic growth factor. And we found that all the stimuli produce led to the production of PLGF inside the spleen, and this was directly related to blood pressure increase. And in the absence of this growth factor, blood pressure was not increased in response to Ang2, salt-sensitive hypertension, or obesity-induced hypertension, suggesting that this neuroimmune response is necessary for the primary onset of hypertension. But in terms of anatomical connection, we were curious about how the brain directly connected the spleen. This is a typical representation of how the inner organ of our body make connection with the brain through the left and right chain of ganglia. These are the sympathetic ganglia, but the spleen was something different, and it was postulated that the connection was mediated by the celiac ganglion that is in the splanchnic district. So this was an idea that a neuroscientist, Kevin Tracy, had from several years, postulating that this was the basis of the cholinergic inflammatory reflex. And in collaboration with their group some years ago, we decided to do this experiment. So we injected an anterograde virus into the dorsal motor nucleus of the vagus nerve of mice that were at the same time injected into the splenic parenchyma with another virus, retrograde virus. Then we collected the celiac ganglion and we analyzed the presence of fibers from the vagus, here in green, and the neuronal bodies of the splenic nerve in M. Sherry here, that co-localized inside the celiac ganglion, suggesting that this is a real anatomic station that connects the brain to the spleen. Why this is important and relevant in translational perspective? Because this is something that we can modulate with bioelectronic tools. So basically what we decided to do some years ago was to find a way to modulate the activity of the nerve at this level, so at the celiac branch of the vagus. We all know that we have stimulators for the cervical vagus, but this is an activity that modulates all the afferent and afferent fibers of the vagus. Here we can directly modulate the vagus that regulate the immune response. And with another electrode, we were able to measure at the same time the activity elicited on the splenic nerve. This is the protocol where we have a vagus nerve stimulation and then the recording of the splenic nerve. And we were able to show that with this protocol in mice that have no stimulus, we can elicit a similar nerve firing and a similar neurotransmitter release, suggesting that we can modulate immune responses in the periphery. But why this is important in cardiovascular disease? Because this is important because we can control what we call the residual inflammatory risk. The residual inflammatory risk is the principle whereby patients that have a good control of their primary risk factors still experience an increased target organ damage and cardiovascular risk. And in the few slides that I will show now, I will give some example of this residual inflammatory risk in the context of atherosclerosis and heart failure mediated by the brain-body communication. In the context of atherosclerosis, we have used the model of APOE knockout mice that basically develop atherosclerosis during aging. So these are atheroprone mice that can develop athero also in the absence of a Western diet just during aging. This is a model that is particularly interesting because it develops plaque in the inner lumen part of the aorta. But the interesting thing is that in this model, it was characterized the presence of ATLO, so artery tertiary lymphoid organs that are lymphoid aggregates on the outer side of the vessel, on the adventitia. This was the finding of some years ago of the group of Andreas Sabenik in Munich that was the starting of our collaboration in the search of neural connection of this ATLO with the brain. So we found that, in fact, besides containing lots of lymphoids and myeloid cells, these tertiary lymphoid organs in the outer side of the aorta contain a dense innervation. This is a staining for a general pan-neuronal marker in the adventitia that shows nerves inside the adventitia of APOE knockout mice that when they have developed atherosclerosis and characterize the different type of fibers that are present in this ATLO, we found both sympathetic and sensory fibers. And these sensory fibers were suggestive of a connection with the brain. So this was the hypothetical model whereby this outer part of the atherosclerotic aorta established connection with the brain. And then we decided to investigate whether the connection is really established And to do this, we used again a viral tracing model, injecting a retrograde virus here in the ATLO of diseased arteries, and then mapping into the brain the regions that were positive for these viral particles. So basically, we found that injecting the virus in these mice, we found a dense positivity for this viral particle in different brain areas, but particularly in the brainstem, where the vagus nerve has its neuronal bodies. So this was very interesting because it was suggestive of a connection, of a sensory connection with brain areas that control the vagus efferent. And in functional terms, more recently, we have also subjected these mice to functional MRI to look whether the map, the anatomical map that we found with viruses was also something functional relevant. And with this connectivity analysis, we found that in the brainstem, as you can see here in the highlighted area, there is a significant difference between the connectivity of wild type and APOE knockout mice, so indicating that this region is really working in a different way. But the question was, is it possible that this is controlling an efferent signal to the immune system? So we decided to monitor over time the progression of pathology with ultrasound. This is the analysis of the aortic arch, the abdominal aorta, and the reconstruction that allow us to monitor not only the plaque inside the aorta, but also this aggregate formation in the outer part. So we used the APOE knockout mice, and we analyzed the efferent activity of the nervous system, measuring the splenic nerve activity and the celiac vagus nerve activity that was increased over time during progression of the disease. So we decided to cut off the celiac ganglion to remove the splenic innervation at this stage. So in the phase where atherosclerosis is starting to manifest and ATLO are starting to appear. We monitored the progression of the disease by ultrasonography, and we found that there was a significant protection in terms of plaque progression and plaque vulnerability in mice that were denervated at this level, so indicating that we were able to modulate the immune response in the spleen and counteract the progression of the disease. And in the next few minutes, I will show another example of the heart and brain communication. So in this bilateral communication, why this is relevant for example, for heart failure. So the heart is another organ that is densely innervated by our peripheral nervous system. This is a cartoon, and this is a typical real staining of how the vagus control the heart. And this is mainly an apparent innervation. And we decided to reconstruct this kind of innervation by injecting a retrograde virus into the left ventricular wall of a mouse during a non-invasive eco-guided session. And we monitored the viral expression into the brain, in particular in the nodus ganglion and in the NTS in the brainstem. And as shown here, you can see that the virus is labeling these areas, suggesting that these are directly connected with the left ventricular walls. And we challenged this kind of innervation with a typical model used for inducing a heart failure in mice. We used the C57BL6J, and we induced the pressure overload by aortic bending. This is the typical preserved adaptive remodeling over time in the C57BL6J that in the last years have been well characterized to develop an adaptive response also in terms of immune modulation. So typically we have in this model a macrophage expansion that is characterized by an increase in monocytes and macrophages that goes in the same direction of a left ventricular adaptive hypertrophy. We hypothesized that this response can be modulated by the neuroimmune interaction. So we analyzed the heart and brain circuit, and as we have shown in terms of anatomical connection, we now challenge this anatomical connection in functional terms, analyzing the neurons that are positively labeled for an early marker of neuronal activation, the CFOS staining, here shown in the brainstem. And we quantified the false positive cells, showing that in the brainstem, pressure overload early increases the activity of this neuron. And in the other part of the circuit, so from the brain to the spleen, we measured the nerve activity at the level of the celiac vagus, at the level of the splenic nerve activity. In both cases, we measured an increased activity of the cholinergic sympathetic reflex on the spleen, suggesting that the brain is recruiting the immune response inside the spleen. But what happens if we interrupt the communication? What happened in terms of cardiac remodeling? We performed the celiac ganglionectomy in mice, and we analyzed the evolution of cardiac remodeling after plaque-induced pressure overload. And we found that mice that do not have an intact brain to spleen innervation develop an early phenotype of heart failure, reducing ejection fraction and showing an impairment of the adaptive remodeling that typically develop in C57 BL6J mice. So indicating that this is an adaptive circuit recruited by the heart and brain circuit to modulate the splenic immune response. So this is the final summary of this data that I've shown, that basically clarify that the interorgan communication in cardiovascular disease is really important, and finding how this neural circuit modulated immune response can be a powerful tool to that can be exploited in therapeutic perspective with bioelectronic tools for modulation, neural modulation. And with this, I would like to thank all the people of that contributed to this result, and our collaborators in this work, and our granting sources. Thank you.
Thank you, Professor Carnevale, for the very fascinating talk. And thank you to all the speakers that stayed in time. And we have now then 15 minutes for discussion. And as I mentioned, we have two discussants, and I leave them the pleasure to lead the discussion and to start with the questions, which I guess are numerous. Please.
So,
But Sebastiano, maybe could we ask if in the audience there are some questions before we ask our own questions? What do you think?
Absolutely, Cinzia. First of all, let me thank on behalf of the working group, these brilliant speakers that made this event very important. And we listened to 3 important talks and highly innovative. And in this regard, I want to thank Professor Ameri and Dr. Bertero for the kind invitation of the speakers and the organization of this meeting. So this is the one of the webinars that the working group of the Italian Society of Cardiology on cellular biology and molecular biology of the heart organizes every year. And I think that also this time it was highly successful. And this is because of fantastic talks, and also because of high number of people that were connected. During the talks, I counted more than 55 persons connected. So I think this is another success, and thank you everyone. So before asking my own question, Cinzia, I want to let you maybe ask if the audience have their own questions. Then please proceed.
Yes, so hello everyone. Just a brief comment that, well, excellent speakers. We have this tradition of having, I think, some excellent webinars with the Italian Society of Cardiology, and this webinar confirms the tradition. I only think that, the people who usually attend these webinars from the Italian Society of Cardiology are sometimes more clinically oriented. So you might be ready to answer probably some questions that are more clinically oriented and less basic science for some aspects. I'll try to, because we have a lot of residents or fellows that are connected, and so we might find that. So I'll ask is there anyone who has questions before we break the ice?
Cinzia, there's a question which is actually not clinical in the chat. So I'm reading it. It's from Azana Yats, and it asks, so the question is, if we rank neural circuits out of 100, how much they have control over immune system compared with molecular factors? This is a question, I guess, primarily for Professor Carnevale, but maybe also Wouter and Leah want to answer.
Maybe I can start trying to answering this challenging question. The question is really relevant because, of course, we have a multitude of neural circuits that control our immune system in a direct way, because I have shown some data that clearly modulated the immune organs, but also in an indirect way. And the connection at the molecular level, as I think the question is suggesting, is interesting, because it has been discovered in very recent years that, for example, the vagus, that we know as one of the main nerves that regulate cardiovascular function, is not a single nerve. So there is a study that at the molecular level characterized the vagus nerve with a complex sequencing of all the tiny fibers that are inside the bundle of the nerve. And it has been shown that, for example, if you have a different pattern of stimulation of the vagus with algorithms, you can elicit the release of different cytokines. So there is a pattern that release interferon gamma, there is a pattern that release TNF, and so on. So this suggests that on the one side that we have a multitude of molecular mechanisms that this neural circuit can elicit, and that at the same time that we can think at neuromodulation to recruit specific molecular pattern. I think that we are just at the beginning of discovering this kind of neuromolecular connection, but that it could be a really important translational perspective, because we can modulate immune system not in a systemic way that usually perturb all our immune responses and can be challenging for responding, for example, to infection. But in this way, we can modulate the immune system in a more tissue selective way.
And to follow up on that answer, I think that's the very promising part of it, because all the therapies that we've discussed, or also the anti-therapies we discussed, all have this systemic effects. And for example, to say if a patient, for example, gets toxicity, for example, from these oncological drugs, then the result is that it needs to be stopped. So then we might say, for example, the heart that we all think is so precious, but it also leads, for example, in my case, the cancer to grow and continue, and leads to have a high impact on this patient's lifespan and also the morbidity. So having this targeted approaches or organ-specific approaches is very interesting with these neural circuits.
So Pietro, waiting for other questions from the audience. I have my own, and maybe they can trigger also further discussion. So because I think all these three talks deserve discussion on important points, I guess. So my first question is for the first talk. So I think I found really fascinating the clinical data regarding the prognostic role of circulating PD-L1 levels, right? Predicting the outcome of heart failure. So my question is basically, what is the mechanism? What is the cellular source of circulating PD-L1 in heart failure patients? And the other question is, what do you think is the role of immune checkpoints in heart repair after myocardial infarction? Because adaptive inflammation is also important.
Yeah, and
can I add a comment to that side? Because I had the same question with a little twist. So
yes.
How do you know that what you see in the blood is the cause and not the effect of heart failure or myocardial infarction? So how can you distinguish which comes first? Do you have preclinical data on that?
So thank you both for this challenging question also. So first of all, I think in the type of heart failure, and especially, for example, with the HFpEF kind of phenotype, I think we all agree upon the role of low-grade inflammation, and that this is part of the disease. And of course, in the HFrEF phenotype, we see it more in the acute phase, that these are more patients that develop, for example, with myocardial infarction. So I believe that the PD-L1, we know from PD-L1 that it can be shed. So we see, for example, if we look at the tumor biology, it's a protective mechanism from the tumors. And I also believe, and this is still open for debate, but I think the data, I haven't showed it here, but it's under review as we speak. Cardiomyocytes themselves can also express PD-L1. And I think,
cool.
and yeah, this is very cool. And they can express it on the surface, but they can also shed it. So and of course the question is, and I think this is very interesting, is if it's related from the cardiac shedding that you pick up in the plasma. I don't know yet. That's the honest answer. Of course you can think, but it's always the hard case with these non-specific biomarkers. Is this coming from the heart or not? I think, and we need some evidence to prove it. For example, after myocardial infarction, does you really have a peak? We see these things. I did some modulation over time after myocardial infarction, for example, with time points. You can see some modulation. The question is that from the heart or not? That's something that further research have to shed light on. I do, from your first question, Sebastiano, I do believe that in the first 7 days or the first 14 days, immune checkpoints play a pivotal role in myocardial infarction. As we now also see from these trials with other anti-inflammatory drugs, the question is, what I'm a little bit scared of is we have some initial studies, for example, with IL-6 inhibitors, and then they could lead, for example, in these animal models to complete cardiac rupture because you were too soon modulating it. So this is a little bit the time window and the amount that you have to tone down the immune response. I think playing with that is going to be what we need to focus on in the near future. But I think people, at least from like 5-10 years ago, got a little scared by these IL-6 trials that at least lead to adverse results instead of positive results.
All right, thank you. So another question for the second talk is, so I think that also very interesting. What's the mechanism? How does interferon gamma affect metabolism, switch down, for example, oxidative phosphorylation? Does it affect direct mitochondrial function? And if yes, how?
So, I mean, this is basically the question that we're trying to answer right now. I wouldn't say that I have a clear answer to this yet. We do have some electron microscopy data from these hearts, and what we see is that the morphology of the mitochondria is changing. It's more like the membrane of the mitochondria seems to be more shrinked, but that's all we have right now. We don't know exactly what is happening, but it seems that it's basically directly affecting the mitochondria in some way. But yeah, otherwise, we have yet to really kind of have this link exactly to why the interferon gamma is making these metabolic switches.
Okay, and the last one for Daniela. Of course, I know Daniela's work very well. It's highly tight, and I think really changes the current knowledge on the paradigm on the development of atherosclerosis. So, as you know, Daniela, atherosclerosis, it's of course is a chronic disease, but then when patients have myocardial infarction, one of the critical mechanisms is atherosclerotic plaque rupture. So, what do you think? Is brain immune interface involved in the destabilization of atherosclerotic plaque also, in addition to the formation of atherosclerotic plaque?
Thank you. This is a very important question. And this, in the work we have already published, we analyzed the long time development of athero without challenging mice with Western diet. We found that when we interrupt the brain to spleen neural circuit, we have reduced the progression of the plaque, but we also have an improved stability of plaque. Now, what we are doing in an ongoing project, we are studying mice with Western diet just to have a fast model. And the other interesting thing in this and the difference with the other model is that mice with Western diet develop inflammation and immune aggregation in the adventitia, but not these tertiary lymphoid organs. So our question will be to invest, because the question that you have is important, is just modulating the formation of plaque or the secondary event. This is something ongoing, and we believe that in the two systems, the aging atero and the challenged atero, there is some difference.
Thank you.
Thank you.
So thank you very much. I have a couple questions too. So for the second speaker, in your model of aging, you know, we have a similar model of aging. And what we see in wild-type mice is that what changes dramatically is the gut microbiota composition, and also there is a very important leakage of bacteria or bacterial fragments within the circulation. So have you thought of exploring this possibility as a trigger of basically what you see?
Yeah, thanks a lot for this really important question. So yeah, I think definitely the microbiota will play a role, but we have some preliminary data that I think it's not just about the microbiota. I think so. So we're lucky that we have a germ-free facility nearby the campus, and we got some aged germ-free mice. We couldn't really look at the cardiac function per se, but we looked at the T cells, and we see that this interferon gamma signature is preserved in the T cells in the aged germ-free mice. The clonal expansion is the same, so at least there is a part that speaks that the part of the T cell function is still preserved even if the mice are lacking bugs. But I think definitely a microbiota will be part of the process for sure. Yeah.
Thank you.
Question.
Yes, go ahead.
So I work on genetic cardiomyopathies, and for example, in ARVC, it's known that inflammation is one of the main early manifestation of the disease. So what do you think about inflammation in cardiomyopathy, genetic cardiomyopathies? Because in this talk, we had an idea about heart failure, atherosclerosis, myocardial infarction, but nobody, really said anything about genetic cardiomyopathies. So what do you think the mechanism is in this case?
Maybe I start. So I think the process is just like, where does it start basically? Is it like the inflammation driving the damage, or the damage driving the inflammation? And I personally believe that it's really context dependent. It's really context dependent, and for some parts, it's the cardiomyocytes themselves that start the process actually, and then this kind of brings in the immune system and it kicks in. But then it's really context dependent, and depending on whether this inflammation in the end would be like beneficial or not. So it depends on the model, whether again we really need the inflammation to resolve a damage that is happening, and then eventually the inflammation needs to recede, or it's really from the kickstart, the inflammation is basically that. For genetic cardiomyopathies, I think, one aspect is that I think the inflammation per se needs a second hit. So we do have a couple of projects here that are trying to look at the desmosomal genetic cardiomyopathies, and what they see that inflammation plays a role. However, it needs a second hit. It needs like a viral infection or something for the immune response to kick in and do something. So I think definitely there is something going on with the inflammation, but depending on it's really disease context dependent in this case.
Yeah, and if I may follow up on that answer, I completely agree. Because we have some data on ACM, let's call it, not RVC anymore, like the ACM phenotype, but also these cardiomyopathies as DCM, HCM, the immune profiling is completely different compared to all these acute diseases. Completely different profile, different mechanism. Don't have to be T-cell driven. It starts maybe with a macrophage somewhere lying around myeloid cells. It's so nothing you can compare with this. You cannot extrapolate anything, at least from my talk, to the ACM phenotype. But it's definitely different, interesting. And so if you, and we can always talk about it later, how to see if we can tease out these immune profiles. But it's completely different.
If I can add one thing that maybe is interesting in this, because the question is, I think is really important. There is a paper published by the group of Cory Lavine and Slava Eppelman. It's an immunity paper of 2-3 years ago, where in a small part of the paper, they show that there is a mechanism of macrophage expansion, resident macrophage expansion, that is shared by a model pressure overload induced by aortic bending and a genetic model of cardiomyopathy. And they show that there is a specific population of cardiac macrophages that respond to IGF that is important in this response of the myocardium in both cases. I think that one of the, in our model, for example, we know that when we induce a pressure overload, we recruit this elicited response. So probably one thing that can be shared also with the genetic cardiomyopathy is that when the myocardium is stressed, it induces some sensory, and when you activate some sensory neurons independently of the cause that is activated, for example, by stretch, by mechanosensing, they will elicit the same recruited response. So this could be a shared mechanism, and we can take information from these models that are more sporadic, not genetic, for investigating more the genetic diseases.
Can I ask the questions?
Sure.
Okay.
Please.
My question is related to sex differences in cardiac immunology. For example, like, either for speaker 2 or any of the speaker 1 or 3. When you see, for example, when you induce the interferon gamma in the heart using the AAV8, practically, I think it's not specific to the cardiac myocyte. So in this case, how much effect of this interferon gamma to the cardiac myocyte? And do you see like the sex differences, how female mice respond to the interferon gamma and how male respond to it? And in terms of treatment, do you think that we should give the same drug or same dosage to male or female patients?
Yeah, thanks a lot for this really, really important question. I think in general, like, unfortunately, the research is, I would say, quite patriarchal so far. It's shifting, of course, and it's becoming better. And we're addressing the question of sex difference more. But there's a lot of questions that are being answered only in male preclinical models, when definitely there is a huge difference between males and females. At least for the kind of baseline physiological aging, we try to address this a bit, and we sequenced both male and female mice. We see that, at least in the mouse models, we see that the immune cells actually were more activated, and this interferon gamma was even more pushed in female mice. However, let's say when we infarct mice, interestingly, we tried also to infarct aged male and female mice, and we see that actually the female aged mice do better, not worse. So it's quite interesting, but unfortunately we don't have like animal approvals to follow up on this yet, but it's definitely something on the table that we want to address. And then for the AAV model, I mean, one part is about the organs. We first thought about having an AAV9 to have it directly targeted to the heart. But then we thought this would not really model what we see with the aging, because with the aging, we really see this interferon gamma signature kind of systemically, and it's really more about an inflammaging phenotype all across the organs. It's varying between the organs, but it's there everywhere. And indeed, we also looked in the liver. We see kind of a similar metabolic shift. However, then we focus on the heart, and we have some echocardiography results that point actually that it's kind of shifting a bit ejection fraction, for instance, a couple of parameters, but not significantly. But regarding the sex difference in this model, unfortunately, we only did this in male mice. We couldn't get approval for both, so unfortunately, this is a question that we've yet to answer. But thanks a lot for the question.
So do we have more time for questions?
Unfortunately, I'm
Moderators.
we have to close.
Oh, that's too bad. But anyway,
yeah,
that means that the meeting was excellent.
Yes, the discussion was as excellent as the talks were, I think, but we are already 10 minutes late. And but I wish to thank all the speakers for their outstanding talks again on behalf also of the working group on molecular and cellular biology of the heart of the Italian Society of Cardiology. And of course, thank also to the discussants and all the participants. I think we learned a lot and we still have a lot to learn on cardiomyology. Thank you again for the...
Thank you.
Thank you very much.
Thank you. Bye-bye.
Thank you.
Bye-bye. Bye-bye to
Bye-bye.
everyone.
Bye. Thank you.